Hi everyone, my name is Aishwarya and I'm a fifth year master's student. I work on a project called Aptamer Pursuit. I work in the Missile Lab and I'm advised by Luis Seze, Jeff Navala, and Kevin Jamison. I'm going to tell you about a project called Aptamer Pursuit. The project I'm working on has to do with two molecules. One is called an aptamer and the other is called a protein. An aptamer is a molecule that binds to another target molecule. In this case, our aptamers are based off DNA nucleotides, so A, T, G, and C, and the target molecule is a protein. The protein in this case is a short amino acid sequence of about length 8. We are trying to predict whether an aptamer and peptide will bind. You can see in the diagram on the right that the protein sequence is long enough so that it has a 2D and 3D structure. However, the peptides in our problem are relatively short sequences, so we only need to look at the primary structure, which is the actual amino acid sequence. We want to understand the relationship between the amino acid sequence of the peptide and the aptamer sequence enough so that we can generate a binding pair. What that means is that given an aptamer, we want to be able to generate a peptide sequence that will bind with it. And given a peptide, we can generate a binding aptamer. This is useful for a variety of reasons. Predicting the behavior of these molecular interactions can be useful for immunotherapy, drug design, and diagnostics, amongst other things. For example, cancer cells emit peptides called neoepitopes. If we know that a particular cancer cell emits a certain kind of neoepitope, we can generate an aptamer sequence that will bind to it. By localizing that neoepitope, we can now introduce drugs and diagnostic measures that will target only that cancer cell. We're fortunate to have this project to be a part of the Missile Lab. The Missile Lab brings together students that are interested in synthetic biology, chemistry, electrical engineering, and computer science. As a result, we also have a wet lab where we can generate the data for this project. However, we can only quantify interactions that happened, not the ones that don't. This means that we only have positive data, which is a set of pairs of aptamers and peptides that do bind. We don't have information about aptamers and peptides that don't bind. We also don't have empirical measures of binding affinity or how well the two molecules bind. So we need to treat binding as a binary feature. They bind or they don't bind. Machine learning models need both positive and negative data. In order to generate negative data that comes from the same distribution as our positive data, we need to first figure out the distribution of our existing data. Then we can generate pairs that do not appear in our dataset. Since we don't know anything about them, we consider those to be our negative examples, those that don't bind. Now we can construct our models. The goal of our initial model is to distinguish between a pair that binds and a pair that does not. As you can see on the right, the input is a peptide and aptamer, and the model outputs a binding affinity prediction, which is some measure of whether the pair binds. The output is a score that doesn't have anything to do with empirical binding affinity measures. If we can perfectly distinguish between a pair that binds and a pair that does not, we've solved our initial problem. So far, we've tried two model structures. The first uses stochastic gradient descent and a neural network to distinguish between one-hot encoded matrices representing the aptamer peptide pairs. We find that this model has trouble distinguishing between binding and non-binding pairs. The second model we've tried is a binary classifier. This works exactly how you would imagine, distinguish between pairs that do bind and pairs that don't bind. This neural network uses the kernels and the convolutional layers to represent an n-gram-like analysis. We find that this binary classifier performs slightly better, but it's difficult to understand whether the patterns and motifs that the network picks up on are biologically relevant. We plan to continue to iterate on these models for a while to improve precision and recall. Feel free to reach out if you have questions. My email is on the first slide. Thanks.